presentation.
How do I do that?
Usually, are you, yes, share again,
It's being recorded now.
and then this one right here.
He gets one more seat.
2 minutes and then.
Well, the good part for Jake comes later, so I've got a lot of slides. We can just go ahead. All right. Good afternoon, everybody. Thanks all for coming. I gave it the title Cancer Therapeutics. I could have just as easily given it this title. Oh, nothing happens. All right. This is going to be a fun afternoon, I can tell you that. Okay, here we go. Let's try that. Oh, that's my other title. There's a lot going on that I want to tell you about. And so it's basically going to be a little bit of a whirlwind tour with like a pre-introduction to the talk. So this first part is just a pre-introduction about, so first thing I want to show you are my disclosures, and I will give you a few seconds to look at them. I'm not going to read them. Okay, we go on to the next slide. So we do translational research on cancer and ultimately want to impact on patients. So a long time in my career, probably 20 years ago or so, I started to learn about the Bayh-Dole Act. And how many people know what that is? All right, so the Bayh-Dole Act, so you remember Bob Dole, came up with this. So before 1980, universities had patents, and for the most part, they were owned by the government because the government funded the research. And nobody really was interested in those technologies for the most part because the government owned it and nobody really wanted to invest in that. So what the Bayh-Dole Act allowed universities to license patents to companies and really for the public good. And so I learned early in my career that this is something we're all supposed to be doing. We're supposed to be thinking about our research and trying to do something to commercialize it and so it can help people in practical ways. And it goes back to this Bayh-Dole Act. And we're all supposed to be doing that. So what is cancer? So we work on cancer. And I made this slide a few months ago when I gave my Faculty to Provost series, just as, a general overview of cancer. So cancer is hundreds of diseases. Normal cells transform, they pile up, they grow as a mass, invade and metastasize. Cancers are like little organs with a blood supply and many different cell types. If caught early, many cancers are cured with surgery. But I think, as is well known, metastases to other organs, for the most part, are what kill people and are hard to treat. Exception are brain tumors. They just grow in that space and they don't really need to metastasize much. Cancer is a disease of aging, but can occur earlier in life when it's inherited or due to certain environmental exposures. Almost half of all cancers are preventable. So if you really want to impact on public health and cancer, prevention, screening and prevention are the way to go. Cancer is treatable. Some of it's curable. We've known that about certain cancers going back decades. Lance Armstrong was cured of testicular cancer that metastasized to the brain. Jimmy Carter was cured of melanoma that metastasized to the brain with immunotherapy and radiation. cancer cells by their nature evolve to more aggressive forms that become resistant to treatments. But we do research because that brings hope for new treatments. So again, by way of introduction, in the United States this year, there'll be almost 1.9 million new cases of cancer and over 600,000 deaths. In Rhode Island, we will have nearly 7,000 new cases of cancer and over 2,000 deaths. Okay, I did something bad. Okay, here we go. So this is taken from a review I put together for ASCO about 4 years ago, and it was about cancer heterogeneity. I'm not going to go through it in detail, but just like kind of look at this in terms of what we're dealing with as far as cancer. So first of all, every clinician is aware of clinical heterogeneity, and if you actually talk to clinicians, they'll tell you, we've been doing personalized medicine the whole time. Because of this clinical heterogeneity, we treat people appropriately for their conditions. And so there are all of these clinical aspects of heterogeneity that you can see there. We're all different as people, as hosts. And the cancers themselves are also very heterogeneous. And so and by the way, for the host heterogeneity, microbiome is there. And that's a new frontier really that we haven't even begun to really understand well and manipulate. But then as far as tumor heterogeneity, there's inter-patient heterogeneity. I think most people are aware of that. But there's intra-patient heterogeneity, and there's intra-tumor heterogeneity. And so intratumoral heterogeneity occurs within primary tumors. It can occur between metastases or even within a metastasis. And a good example a few years ago was a New England Journal paper in kidney cancer where they took multiple biopsies of the primary tumor and the metastasis, and it took 4 patients to get a New England Journal paper, but that was about 10 years ago. I think people still are doing that with the right type of study. The tumor microenvironment is incredibly heterogeneous. There's immune heterogeneity, stromal heterogeneity, stem cell heterogeneity. There's geography with low pH and hypoxia and altered metabolism. And all of these things are going on. And then there's, heterogeneity in the response to treatments as tumors evolve. And there's a powerful selection pressure with therapeutics. So in a way, that is the opportunity. If we want to do something to address the problem of cancer, we need to be able to address this complexity. So it's not really all very depressing. I mean, I think if you look at the hallmarks of cancer, there's a finite number of hallmarks, certain pathways and certain aspects of the tumors, they're all there. Then, of course, there's the immune system. So, if you were to ask me, are we ever going to cure cancer? I'll say, well, we've already cured some cancers, but to have a chance to really have impact, we need to harness the immune system because, the T cells and other cells that attack cancer, they're like our soldiers that are going out there, and they're really good at what they do, but we have to help them sometimes because they aren't working as well. So just a word about, what are some of our tools right now for precision oncology that the clinicians have? We can certainly analyze tumors. I mean, this is a pathology department, so this is what pathologists do in terms of histology of the tumors and some genomic sequencing, and are genes amplified or deleted, and is the tumor proliferating? And this is stuff that pathologists do every day. We also have blood biomarkers, and there's a whole bunch of them, as you can see here. As far as the host, there are susceptibilities to cancer, but there are also things that are just still not well understood. All the thousands of polymorphisms that predispose to certain diseases. We have serial monitoring, and our capabilities are getting to a point where we can do serial monitoring of cancer, and I'm happy to talk about that. There are various databases and opportunities to mine genomic data with clinical outcomes. And we have laboratory models such as patient-derived xenografts, organoids. In the clinic, we have genomic tumor boards that have cropped up over the last few years to help make sense out of all of this information, where it's not always obvious what to do with it. And, this is an area of expertise that is still kind of developing in terms of how do we train people to be good at this type of work, on the clinical side. So a bit about my career, and I'm seeing some new faces perhaps in the audience who may not know. This is about my own journey here. I was a MD PhD student in Miami. Then I did a residency and fellowship at Johns Hopkins, and then joined the faculty at UPenn, and then moved a couple of times, Penn State, Fox Chase, Temple, and then moved to Brown about 3 years ago. Some of the details in here are specifically for Jake, so he can see that since we moved here, our citations are going up. Earlier when I was at Penn, they were flat for a very long time. And there's a message, if your citations are flat for a long time or going down, you should look at that and try to figure out what to do about it. Publications in Pubmed. So 2021 is a really good year for us. It's the best year ever. You always got to balance quality with quantity. So I think we have quantity this year and a little bit of quality. Hopefully we can, reach some optimum of both moving forward. All right, so that was the pre-introduction to the actual talk. So this is what I'm really going to talk to you about. The early years, some key discoveries and their impact. What we're kind of doing here as far as research at Brown. And then right at the end, a little bit about the Legorreta Cancer Center, which is being, our cancer center is being renamed after a big gift. And so I thought I would just show at least one slide about that. So some of these slides are going to go by, and I'm not going to say much. I have a big family, and this is part of the family. We got together in September this year in New Jersey because my aunt Susie, who's right here, was celebrating her 50th wedding anniversary. And there are a lot of doctors in the family, and I've always been the outlier. I have to kind of explain to people what it is that I do, why I'm not a regular doctor that just spends all his time seeing patients. And but within this really large family, and really both sides, maternal and paternal, there are enough doctors to basically run a hospital, maybe more than one, which is kind of interesting when we get together, some of the conversations. But I found this on the internet. This was made by an uncle of mine who lives in Canada. And I recognize some of the faces. This is my grandfather, who's a accomplished physician in the Middle East and in Egypt. And this is my grandmother, and this is my mother. So when I looked at this, I thought, oh, okay, this has got to be like somewhere in the 1950s. But then, as I looked more into it, I saw this guy who is apparently my grandfather's father. So what I figured was going on here was my uncle kind of made this up. This is Photoshop. And these things are called, Attila will appreciate this, tarbushes, which are Turkish, right? But I don't think we're Turkish, but I have no idea why he did this, but I saw it. I'll ask him the next time I see him. So growing up overseas before my family moved here, I went to this British school in Alexandria, which I looked it up yesterday. You know, I'm pretty sure I know this is the school I went to. It's very hard to actually remember what the building looked like, but it was definitely a British school. We lived on the same street as the school. So on the one side, I could see the school from, you know, the 5th floor where we were living. And on the other side, all the way over there, we could see the Mediterranean in this building that my grandfather built when he moved from the South. And as far as I could tell, the school was started in 1902, and it was sort of an elite school, which I had no idea when I was a kid going there. Apparently, some famous people have gone to this school. So I looked up, they actually have a Wikipedia page. So yesterday I added myself to the Wikipedia page. I was going to show you that, but all right. So that same uncle worked out a genealogy of the family. And so these are my parents and my younger brother, probably in the early 70s. This is what I looked like in the 60s, and this is with my paternal grandmother. And within that genealogy on the maternal side, these are my grandparents, parents. This is me, Evelyn, and our 4 children here. You know, it's just interesting to see this put together. My family used to always go to the beach in the summer because many of them lived in the South, and so they'd come and we'd go to the beach with them. As an aside, I have a lot of sunburn from those early years because we didn't use sunscreen and we just kind of stayed there in the sun, you know, just getting burned. And I've asked some people who work on melanoma, you know, do you have a way of like knowing what a person's been exposed to, you know, for personal reasons? I'd like to know, am I ever going to have melanoma? But nobody could tell me. It's like they can't like figure that out. My uncle, this is that same uncle, has a villa on the Mediterranean, and I guess at some point I went to visit him. I do remember we had a barbecue that was very nice. So I went to Martin Van Buren High School in New York and Coral Gables Senior High in Miami. So how many people here know who Martin Van Buren is? Okay, so I gather you know he was a president. Which president was he? Good answer, Dan Spade. That's right. So, you know, I would know that because I went to that high school. Yeah, so, and the Coral Gables High School, our football team was known as the Cavaliers. That's when I learned what the word Cavalier means, so I had to look it up at that point. They played football. Well, football is really big in Florida at the college level as well as the high school level. So when I was in high school and college, I worked at the University of Miami. They had a gifted program. And so Dr. Awad was my first mentor. He was actually, you know, I was looking this stuff up about him. I certainly didn't know all these things that are listed here, which are very interesting about him. He was the director of the MD PhD program in Miami. I worked in his lab in high school and in college. I did end up in the MD PhD program. But this is where my first exposure to biochemistry. So what did we do back in the late 70s? Well, they would operate on patients and we would get like their intestines in a bucket or their placenta. And, you know, we would make proteins out of that, you know, spent a lot of time in the cold room, running columns and pipetting by mouth. I know what DMSO tastes like. It does not taste good. So he was a great role model. He was a physician scientist himself who was a hematologist. He passed away a few years ago. I remember one time I stayed at his house in Coral Gables while he and his wife were on vacation. And I remember another time as an MD PhD student, like the university had a museum and they had a wine reception, and I got to be the bartender at that event, which was fun. So my thesis advisor was Dr. So, who's originally from the Philippines, and his contribution, so he was a very accomplished physician scientist who was at some point in his career an American Heart Association established investigator, and then a Howard Hughes investigator, and then an American Society for Clinical Investigation member. But he discovered DNA polymerase delta doing biochemistry on calf thymus mostly, but also with human placenta and other tissues. And he, while I was in the lab, but I didn't get to work on that project, but it was a good project, he isolated pCNA. So, you know, we were doing these DNA synthesis assays where you can see how long the DNA is when it copies, and there's a factor biochemically purified that made you copy long pieces. So it was known as a processivity factor because it was doing that. Processive DNA synthesis versus distributive DNA synthesis, where it just falls off and doesn't copy long strands. And, you know, that was pCNA. That's one of the ways that it was discovered. Bruce Stillman, who's the head at Cold Spring Harbor, also discovered it around the same time. And anyways, Dr. So was a great role model. He was also a hematologist. When I was a student, I had the opportunity of meeting Dr. Arthur Kornberg of Nobel Prize fame. We invited him to speak, and so I got to hang out with him for a day. You know, his prize was in the field of DNA replication. And Dr. So and his family owned a really nice Chinese restaurant in South Florida called the New Chinatown. I'm pretty sure it's got to still be there, but it was great. We had many nice lab dinners there. We also used to play tennis on the weekends. You know, he was good. The lab was co-mentored by another senior person named Dr. Downey, and we had a postdoc, and so the four of us would play tennis on weekends, and they were all much better than me, I'll tell you that. So I moved to Johns Hopkins and for my residency in medicine, and Dr. Jack Stobo, who's a well-known immunologist, was the chair of medicine, and in my third year of residency allowed me to take a research elective. And so I got to work with Dr. Balin in his lab. This was around 1989. So I set up the first PCR machine in his lab, which was fun. You know, PCR, you had to hook it up to the tap so that you could cool between the cycles. And I used the PCR to clone a fragment of the first human DNA methyltransferase that was cloned, and used it to do basically northern blots on progression stages of colon cancer. This was published in PNAS, and it was communicated by Dr. Victor McCusick, who's very well-known human geneticist and founder of the HUGO, the Human Genome Organization. And so I could have stayed in the field of epigenetics, but I'm not going to say the story publicly here about why I didn't stay. But sometimes certain interactions in certain labs kind of, you know, the lab isn't big enough for a number of people, and you just say, well, you know, maybe I can work somewhere else and maybe have a better chance. And so I sought out Vogelstein's lab. And at the time, you know, I mean, he was very well known for colon cancer genetics and for the Vogelgram. And how did I first hear about him? You know, when I was a resident, this was during residency, I subscribed to the New England Journal. It's the only journal I subscribed to. And one day there was an issue, and it talked about the multi-step nature of colon cancer. And basically they were getting pieces of polyps or larger polyps, small polyps, normal epithelium, from patients by having, a GI doctor collaborate with a pathologist who's collaborating with a geneticist. And they basically, that was the early version of the Vogel grant. Like the more advanced stages just had more mutations. So they ordered the events based on that. And so Bert didn't make it easy to join his lab, and that's a story for another time, but I persisted, and I'm very grateful for the opportunity to have worked with him. When I first joined his lab, the P53 project was ongoing. It was, they had just recognized a couple of years earlier that it's really a tumor suppressor gene, not an oncogene. So for 10 years, Cell Science and Nature, everybody talked about it as an oncogene because everybody was studying the mutated P53. And his lab figured out that, oh, it's really a tumor suppressor gene. And when you introduce it into cells, you suppress their growth, and that everybody was working with the mutant version. So when I joined his lab, it was known to be a DNA binding protein that could bind non-specifically as well as specifically, but it wasn't clear what it recognized. So he said to me, well, go figure out what it can bind to. So if I show you a DNA sequence, you'll tell me whether it binds or doesn't bind. So we did that. And that paper was published in the first issue of Nature Genetics in 1992 on page 45. And that was a consolation prize because Nature wouldn't accept that paper. But it was the right sequence. And it's a story for another time exactly about how we figured out that sequence by aligning all kinds of sites that were immunoprecipitated and sequenced. So after that finding, we thought we had a way to discover genes that might be regulated by P53 that could suppress cancer. So then pursued genes that are regulated, but there's something that happens when you're in a research lab where your mentor wants you to do something and something you really maybe don't want to do, and you're reluctant to do it. Like he wanted me to do a chromosomal walk. Okay, so I don't know if anybody here knows what that is, but you've got a piece of DNA over here, and you've got a gene over here, and you basically make probes and kind of keep marching down and discover a gene that's near a sequence that a transcription factor binds to. And I just felt like, I'm in a box here. If I didn't IP those sites, I'm never going to find anything important. So I, dragged my feet and just didn't want to do that. And then along came a new technology to do subtractive hybridization. And that was before microarrays or anything like that. So we could look at differential gene expression. And so I said, yeah, I'll give that a shot. So I did. I made cDNA libraries from a glioblastoma cell line with and without P53 and cloned P21. And it was easy because it's a small gene and it just kept coming up over and over. And it was clearly differentially expressed. So, Bert's been great. He's famous for telling people what's next. You've made a big discovery. What's next? The other thing, well, there are many, many things that happened in that lab, but one of the memories I got last night was one time I did 200 mini preps all at once. And I did them during lunchtime because that was the only time all the centrifuges were available. And I remember myself running from bay to bay because some of the steps have, they're not long spins and you just have to go and get the tubes out and do things with them. So that was fun. Later on in my career, when I mentioned that to somebody, I think maybe on social media, they were like, well, it was probably a lot of contamination. All right, thank you very much. I'm sure I got my clones at the time. So anyway, so this is P21's discovery and its role in cell cycle regulation after DNA damage. And for years, I thought this would be the most important research I would ever do, which was kind of a depressing thought, but it's not really true. And so Jake wasn't here for the first version of this, where you know, the timeline of working in different institutions and citations by year as a faculty member and numbers of papers published by year. And so I would say the real fun began after starting my faculty position at the University of Pennsylvania. And just to repeat for Jake, this can happen sometimes where your citations are flat for a long time, and then you know it's probably time to move, go somewhere else. And this is what's happening here at Brown, so I'm very excited about that. You could see in numbers of papers per year, there's a little bit of a roller coaster, and this is not by accident. This can happen too as your funding goes up and down. And but anyways, it was a lot of fun. And so up on the 5th floor, if you walk through the lab, you'll see this thing with the different pictures of the lab since it was established in '94. And I tried to do like a pano of that, like walking across. And, you know, this is the best I could do with that. You see how some of the pictures are wavy, but the lab has always been very international. And I don't, this probably is too small, but this map, which there's a physical map in the lab with where everybody has come from over the years. And it's one of the really fun things about the lab is just how diverse it is with lab members from many parts of the world. I remember one guy many years ago, I won't mention his name, but well, maybe somebody will figure it out. He was writing his lab notebook in Russian. And I basically had to tell him, no, you can't do that. Got to be able to read it. One of the aspects of this lab that I think has helped all along has been the idea of bringing new technology into the lab and embracing that and seeing what could we do with it. So, you know, over here, you know, I forgot to say this earlier, but I do have my green laser pointer, which isn't working. That's really, it was working before. Okay, well, anyways, this Xenogen machine there was the first one that was bought in Philadelphia in 2002. It was a gift from the Howard Hughes Medical Institute, you know, where the justification was, I need a Xenogen to do mouse imaging. And, you know, the instrument would arrive, and it still works. It's moved everywhere we've moved. We acquired this robot a few years ago through a U-54 that we had in the field of molecular imaging. And I've done a lot of drug screening with this robot, which can do 25,000 compounds a day. And then over on the right there is our hypoxia chamber. We have a hypoxia chamber. We've had one since the mid-90s. It breaks down frequently, and we're having to fix it a lot, but it's important because hypoxia is a major issue for drug resistance. So before coming to Brown, this was usually my last slide where you know, I'd show pictures of the lab and the funding over the years. And then we came here. And but in terms of new technologies, you know, so, you know, we discovered this death receptor, which I'll mention a little bit later. You know, we were the first to sort of combine bioluminescence imaging with gene knockdown in vivo to demonstrate the growth advantage when you silence this death receptor. We were among the first, I can't really prove this, but I think we were among the first, I'll say, to do molecular imaging for drug screening going back a long time. We did have some forays into vascular imaging, and this is some of what you can do. This is an unstained tumor. This is the tumor itself. There's a spectral shift within the blood vessels with spectral unmixing, you can get images like that. And we did publish a paper in Biotechniques years ago about how you can get a vascularity index by doing this. And we did other things. Whoops, we do have this, okay, is this gonna work? The flow cytometer, which doesn't work. Oh, here it is. So this is a flow cytometer from the mid 90s, and I was the only one left who could work it before it died over the summer. And you know, we did a lot of our cell death assays with the flow cytometer. When we got this flow cytometer, you know, it's another gift from Howard Hughes. We need a flow cytometer to do flow cytometry, and here it came. And interestingly, back in those days, most facilities did cell surface markers. They didn't do cell cycle, and that was one of our interests. And so we developed a lot of protocols for cell cycle and cell death. This is one of the things we did with the imaging. So at one point, we transformed human esophageal epithelial cells with different oncogenes and created tumors, in which you can see from this little movie, which probably went by too fast, is that different oncogenes drive cancer growth at different rates. And so this tool is definitely available. I mean, this was 15 years ago that this was possible. And you could do, you know, gene expression profiles, and these were of esophageal origin. And so some of the transforming oncogenes gave gene signatures similar to adenocarcinoma, while others to squamous cancer. And so you could recreate models like this. So, you know, questions we've been asking, as I think most of you know, how do cells die? How do tumor suppressors like P53 cause cell death? How do cancer cells evade chemo and the immune system? What are cancer's vulnerabilities if we're going to develop new therapeutics? So, you know, some of these are going to go by very quickly because there isn't that much time left. So, you know, the discovery of death receptor 5, this was subtractive hybridization, and we created a knockout mouse that had deficiency in cell death after radiation, and also had inflammatory lesions in the lungs due to a deficiency in the trail pathway. We're doing some experiments right now actually to try and restore trail either by a small molecule or actual trail infusions to try and address this clinically important problem. This is a movie I love to show about trail, you know, and it's only 20 seconds, but these are lung cancer cells, what happens to them over 4 hours as they're treated by trail. This is why we like this pathway. It's part of our innate immune system that suppresses cancer. And it's, you know, there's no doubt what's happening to these cells. And so why doesn't this work well in humans? And what can we do to get this pathway to translate? So, you know, so we do drug screens, and then we try to work our way towards clinical trials. We discovered this drug, ONC201, and published a couple of papers about its mechanism, which are discovered as a trail-inducing compound, because we thought, well, that would be a good way to induce trail, to do what I just showed you. And so initially, we figured out a mechanism where dual blockade of ERK and AKT would release FOXO3A to go to the nucleus and activate the trail gene. And then later, we uncovered activation of the integrated stress response through some EIF2 alpha kinases that are upstream of ATF4 CHOP. And interestingly, regulation of death receptor 5, the very, there we go. It's working now. I don't know what happened before. Sometimes when you need it to work, it doesn't work. The drug has an activity against cancer stem cells, both in culture as well as in vivo, whereas classical chemotherapy actually enriches for cancer stem cells, and essentially through the same mechanism, and you could show this in 3D cultures as well. The drug entered clinical trials and has a signal in various tumor types, including endometrial cancer and prostate cancer. We published a paper about 3 years ago now showing an anti-metastasis effect as well as immune stimulation, in this case of natural killer cells. So this is a cool system where the tumor cells are green, the live cells are blue, the dead cells are red, and you could sort of merge the images. And so the natural killer cells in the presence of the drug are killing the tumor cells. And you could reduce that a little bit by an antibody that binds to TRAIL. That's the RIC2 antibody. This is in prostate cancer. In the first in-human trial, there were 8 patients with prostate cancer, and 4 of them stayed on the drug for more than 6 months. There's never been a clinical trial, a phase 2 trial in prostate cancer. We're interested in pursuing this. A couple of these images down here, oh, now it's not working yet, are from men who are over the age of 90 who tolerated this pill, and their lesions got smaller, bone metastases or visceral lesions. And on the left are immune signatures that track with responders versus those who had a short progression-free survival. So the immune stimulation appeared to correlate with a longer progression-free survival. We have published some other work from the lab showing anti-tumor efficacy in mouse models, and have been working on a protocol combining the drug with hormonal therapy and other agents, including enzalutamide and more recently with darolutamide. Now, there was a first clinical trial in patients with brain cancer that was done at MGH, and it enrolled 17 patients. And of those 17, a couple of patients had exceptional responses, and they upregulated markers of ATF4 and DR5 and showed evidence of cell death. And this is a 12-year-old girl who had these lesions in the middle of her brain and couldn't smile, and over a few months could regain her smile. And so it turned out that those 2 patients had H3K27M mutations. And so at this point in time, there have been 50 patients with H3K27M mutated diffuse midline gliomas. And the overall response rate is about 20%. So those are they're very strict criteria. So in solid tumors, it's usually 30%. In brain tumors, they want to see 50% shrinkage of tumor before they even call it a response. But then there's also stable disease in some of these patients. There is ongoing work to try and understand why some people just do not respond. Now, this is another patient whose tumor basically shrunk. This woman was treated at Dana-Farber, and her tumor responded very well. She was able to go back to college, and a few others have had partial responses. And it's thought that the median survival of this population is significantly greater than the median survival of these diffuse midline gliomas. So there is prolongation of median survival. So with regard to these mutations, we're taking 3 directions in the lab, combining this drug with EZH2 inhibitors. And this is a direction we pursued because EZH2 methylates H3K27, and we thought if we could inhibit the methylase, maybe this will mimic the mutation, and so that proved to be the case. Yes.
Do you have to worry when you take that approach to reduction decrease it?
Well, I mean, we've done the experiments, and the story actually got very more complicated. We published papers this summer about what's happening at H3K27 methylation, acetylation, and synergy. So the fact is you see synergy. Interestingly for me, didn't necessarily expect this, even though I did some work in epigenetics long ago, I wouldn't have predicted that the HDAC inhibitors would be synergized better. And from our work done by Yikun, working with Attila, has shown that there is some correlation with really the you end up acetylating H3 K27 as a signal that does associate with synergy and response. And, you know, we're trying to see what we can do in the clinic. Another combination we've been working on is ONC201 plus temozolomide plus radiation. And Lanland's been doing this work now for a few years, and including the last year doing some in vivo studies. And so, you know, it was the summer of 2021, and we got together for the first time in about a year and a half. And this is the greater lab group that includes collaborating faculty, and this is over the bridge. And this was a very nice day. And I wanted, I don't know, have people seen this? This happened here at Ship Street in May, around 9 p.m. Hmm? Yeah, no, there was something going on and I needed to be here. So I thought I'd take that and share it with you. They do this frequently. Okay, so Chimerix that bought out Oncoceutics visited with us here in July, their leadership, and we hosted a symposium and made 16 presentations by different people pursuing different directions that are potentially translational. Some are very similar, but involving different tumor types, and some involve an analog of ONC201. And so this was a good day of exchange. And some of the key questions, you know, we're continuing to work on the mechanism of action. How could we get the drug to work better in brain cancer? How could we get it to work better in other tumors? What are the mechanisms of the analogs? How can we translate some of this? What are some useful biomarkers? What are resistance mechanisms? And so, you know, cell death is complicated. Benny Carnero and I wrote this review in Nature Reviews Clinical Oncology. And these pathways are very relevant to drug resistance. And the therapeutics we use also, there are a number of therapeutics that target this pathway in different places that have been under development. So it's always a challenge, you know, when you bring in these targeted agents with classical therapeutics. So getting back to the drug after that little break. So, you know, we're working on several disease areas, and I won't talk too much about this, but this was essentially reflective of the presentations we made. This is the orthotopic implantation that Lanland has set up in the lab, along with the second experiment. I still didn't have time to pull in the third experiment, where the triple combination is better than any of the doublets with radiation and temozolomide. And you could sort of see what that looks like. They're hard experiments whenever you have a triple combination and you want to show synergy, because a couple of times the doublets just work too well. It's tricky to get that to work, but I think we nailed it, and hopefully we'll be putting a paper together, resubmitting an R1 that went in, but we weren't ready at the time. So with regard to the HDAC inhibitor and EZH2 inhibitor, Yikun Zhang published in Neoplasia this summer results that I was describing. And so this is, we have this grant from the Warren Alpert Foundation that is trying to bring ONC212, which is an analog of 201, into a first-in-human trial. We're collaborating with Nabil Bardisi at MGH and Dave Tuvison at Cold Spring Harbor. He's also the current president of the AACR, and Howard Safran at Lifespan. And basically, this analog is 10 times more potent. We did some preclinical studies it worked better than the parent drug in every case. And it worked in some cases where the parent drug didn't work in pancreatic cancer xenografts and PDX models. We did that work at Fox Chase. And basically the grant is going to get us to the first in human trial. And we're kind of in the middle of it right now with IND enabling studies to be able to hopefully get to that trial maybe in another year or a little more than a year. This is the protocol schema, which I will not go through other than to say there's a phase 1 and a phase 2 portion with dose expansion and various biomarkers that we will be measuring. And so Alex Raufi, who's a junior faculty in Hem/Onc, is looking at other combos in pancreatic cancer focused on autophagy. So there's good data that autophagy is a very good target in pancreatic cancer, and he's interested in that, combining with MEK inhibitors as well as immunotherapy, and actually has an active clinical trial, not with this drug, but with a MEK inhibitor, an autophagy inhibitor, and immunotherapy at Lifespan. Wenyi Chang is looking at sarcomas, and there are some parallels with epigenetic regulation in that tumor type. And she has some promising synergy data and needs a little more protected time to be able to do in vivo studies and move some of this to the clinic. So over the summer, we had this lab picnic. That was fun in Barrington, Rhode Island. And this is about a 20-second video, but it was nice. It was like the first time we actually got together as a lab since moving here. And we usually do this every summer. And these are some of the pictures you see here of the event. And more importantly, there's our fearless leader. Got to show this because he looks happy. He looks happy. Yeah, he does. You know, and over the summer, I learned how to captain a boat. So this is on Narragansett Bay.
He invited you to this, and I wisely decided not to.
I'm told I'm good. Well, so.
You know how many deaths from motorboats in Barrington?
All right, you see, this is the kind of thing you could see in Barrington. So this is at Barrington Beach. You know, this is a windsurfer. And, you know, I tried windsurfing when I was younger, but I mean, they look like they're floating above the water, which I don't understand.
It's a kite surfer.
Oh, kite surfer. Well, you know, yeah, you're right. You're right. Okay, so, you know, we do have scientific discussions and we do some grant writing. So notice here, there is a laptop on that boat. There's a certain restaurant in Warren called The Wharf. I'm sure everybody knows it, but you can get this cake called Death by Chocolate. Okay, highly recommended. And it was great to welcome Sean and Sheldon and Abbas and Attila over here. I'm sorry, Attila, you got wet that day. I can't remember what did it. Yeah, right. And so if the boat goes in a certain direction, all the water comes on you. And so sorry about that. Okay, so another thing I did this summer was read some books and wrote about it for the Cancer Letter. And these are some of the books I read by either people I knew or was interested in reading about these books. And if anybody's interested in the book reviews, I'm happy to share them with you. Off Our Chests was written by John and Liza Marshall, and they were here visiting with us in the last couple of weeks, and it was a fun day. She's a breast cancer survivor. He's head of Hem/Onc at Georgetown, and it was just a great experience to have them actually tell their story and answer questions and meet with people. So that was fun. I went as far as Newport, Rhode Island, and this is the Newport Bridge. It's 92 feet deep there, but right around the corner here, it gets really shallow really quickly. And if you don't know that, you can hit the rocks, okay? And so if you ever hit the rocks, raise the motor instantly. You got to have that wired in your brain. Don't sit there and watch that rotor just go round and round, because then you're going to have to pay for it to fix it. And so this is what's on my reading list now. I actually started reading a couple of these. Henrietta Lacks, this is by one of her descendants, is a really good book from the family's perspective. And this New Deal for Cancer looks like a really good book, edited by Charles Fuchs, who is just the outgoing director of the Cancer Center at Yale. And it's written by a bunch of people we respect, and there's some really, really good stuff in there. So I'm looking forward to reading all of these at some point. Now, so this was this year's...
Such a beautiful day for a bike ride for the roadie to raise money to support BRUAG, the Brown University Oncology group that is doing innovative clinical trials research for patients with cancer in the state of Rhode Island.
Okay, so that's Bill Sykoff, Care New England, leading breast oncologist. I didn't fall here. And this is Bill and his wife. And by the way, you can all get that t-shirt from AAAS that says "Listen to Science." It's a really cool t-shirt. Okay, so then I fell because I was going downhill and we were lost. So the GPS was here. I hit the brakes and that happened. About a few weeks ago, we set up for the first time in the state of Rhode Island single-cell cytokine profiling through this new instrument we have here, which is very nice. Liz graduated, published her work in eLife, and gave me a bobblehead as a going away present. This is Kelsey, and I wanted to, we're very proud of Kelsey for winning the Women in Cancer Research Scholar Award from the AACR, and I just want to show you a quick video that she made. It's like literally less than a minute. The problem here is that the computer really takes its time. Oh, here we go. Okay, not too bad. Not that one. Okay, here we go. Can you see Kelsey?
Hi, my name is Kelsey Huntington, and I'm a second-year pathobiology PhD student at Brown University in Providence, Rhode Island. My research in Dr. Wafik El-Deiry's lab involves discovering ways to therapeutically modulate the tumor microenvironment to promote anti-tumor immunity. Such therapy could increase efficacy of checkpoint blockade by recruitment and activation of immune cells by a modulation of cytokine, chemokine, and growth factor profiles. After my PhD, I hope to impact the field of cancer drug discovery by developing novel treatments for patients with liquid or solid tumors. I'd like to thank the American Association of Cancer Research and the Women in Cancer Research Council for awarding me with the 2021 Women in Cancer Research Scholar Award. Thank you.
All right, very good. And we're very proud of her. And she got a great score on her F31, which I'm saying after Jake left. Okay, so I have to figure out how to get back here. All right, PowerPoint, okay. And then, okay. Yeah, I might skip the other one where I have to log out because it takes too long. But okay, I see it there. I don't see it here, hopefully. Okay, here we go. All right. We are doing some really cool work in hypoxia, which is, as I mentioned, I showed you the Hypoxia Chamber earlier. So Shui Zhao graduated with her PhD, and she just published this paper in Scientific Reports about a new strategy to target HIF-1 alpha independent of VHL. And it just literally came out in the last couple of weeks. And we have a clinical protocol that is ready to open with this drug combination in patients with breast cancer who have progressed on CDK4 and 6 inhibitors, which are used as standard of care for patients with estrogen receptor positive HER2 negative breast cancer, as well as patients with RB deficiency. This is the protocol. I'm not going to go through it. But Shui also published another paper that just discovered the mechanism of action by which CDK inhibitors destabilize HIF-1 alpha through SMURF, and this involves SMURF2. This involved IP proteomic mass spec where the IPs were enriched for SMURF2 only if the cells were treated with CDK4 and 6 inhibitors. So this was published, and this was a good issue of Oncotarget where she made the cover. She was the cover paper. The journal makes these videos about their papers, but since I'm not going to have time to talk about them, Kelsey had a paper doing cytokine profiling in response to small molecule drugs. We had a summer student who found some really interesting effects of BRCA1 expression and as a function of tumor type. And Lindsay has been looking at transcriptomic and cytokine responses after treatment with chemotherapeutic drugs. This is from that BRCA paper, differences in clinical outcomes between patients with colon cancer and breast cancer, depending on the BRCA1 mRNA expression, which is very interesting, not really well understood, tissue specificity, and maybe it has some therapeutic implications. I think people know that young patients, younger people are coming down with colon cancer. So this work identified that young people tend to have higher expression of BRCA1 in TCGA in their tumors. Again, for reasons that we do not understand. And so this is kind of opening up some questions that we will be pursuing. I will not talk about this other than to tell you this was just published. My favorite gene that I showed you many, many slides ago is not mutated in most cancers, but it is mutated in bladder cancer, which is common in the state of Rhode Island. They're truncating mutations, which is unusual. You don't see those mutations in lung cancer, which is also a smoking-related cancer. And in this paper, we discovered that there is coexistence of truncating P21 mutations in bladder cancer coexisting with RB truncating mutations. And we think that this is telling us something about environmental carcinogenesis because the APOBEC signature is common in bladder cancer, but the APOBEC signature shouldn't give you truncating mutations. So we think there's some carcinogen, or maybe it's still related to tobacco, but it concentrates in the urine, and then it causes mutations in DNA repair genes that then ultimately cause truncations that you would see from frame shifts, or it could be that some of these carcinogens directly intercalate into the DNA and cause frame shifts that way. But anyways, mostly hypothesis generating at this point. Another really notable advance was we just published this first humanized mouse model of cancer that is available to test immunotherapy in combination with other agents. And we really developed this because patients who get immunotherapy have this phenotype of hyperprogression. So they get immunotherapy, their tumors actually grow faster, and there's no mouse model. So Ilyas Sahin, who is a fellow in Hem-Onc, and he just moved to University of Florida for his faculty position, basically worked on this model that has human tumor cells and T cells, and some human tumor cells grow faster when you treat the mice with immune checkpoint therapy. So this is going to be very useful, and this is from the paper showing higher proliferation within those hyperprogressive tumors and altered cytokine profiles that we are trying to make sense of. We just submitted a U54 to the NCI that has brought our group together with John Sedevy's group and Michelle Dawson and Benny Carnero, looking at senescence in the tumor microenvironment. And the one thing that I learned from this is that the chemotherapy drugs we use cause senescence. DNA damaging agents cause senescence. PARP inhibitors cause senescence within the tumor microenvironment, within the stroma, within the fibroblasts. And they also, the senescent cells have altered cytokine signatures, and they affect the response to treatment. So we have organized this group that is basically looking at different aspects of the tumor microenvironment and trying to develop therapy combinations based on the cytokine profiles that we are learning from, and we're combining senolytic therapy with cytokine therapy in this context, and also looking at effects of drugs on tumor cells within the microenvironment that also cause senescence. So this is your typical organization for such a proposal. This is some of the data in the grant, but one nice thing that we did was we managed to sneak in there some single cell cytokine profiling data within the grant, including the Tisney plots and the polyfunctionality score index that looks at the number of cytokines that are produced by single cells, and looking at drug synergies to overcome this senescence. Using these tools, we're looking also at the effects of the therapeutics we're trying to use to address the microenvironment, looking at their effects on T cells and NK cells killing tumor cells. Sheng Liang back there has developed a model where if you inject mice with tumor cells and senescent cells, they grow faster. And so we think this is a very useful model. We're very interested in doing similar things with the humanized mouse model. And this is more drug synergies with the agents that are inducing senescence, and then some insights in terms of what to do after they progress, perhaps due to senescence in the tumor microenvironment. We have a organoid single cell cytokine profiling core, which I'm happy to tell you more about. I don't have time to talk about P53 in this talk, but just understand that we have a strong effort trying to develop small molecules that are active in P53 mutated cancers, and we're recognizing a couple of very important mechanisms. One involves P73, which is stimulated by small molecules. The other involves the integrated stress response, which activates pro-apoptotic P53 targets that have anti-tumor effects. And we have become very interested in this mechanism as a plausible way that you could restore a tumor suppressive pathway to achieve an anti-tumor effect. This is some of our directions with organoids, and you could just take a look at that. I'm not gonna, I think one of the exciting areas is transgenic organoids with inducible alleles, where you could take tissue out of the mouse, induce the alleles that are relevant to tumor progression, and then ask a bunch of questions related to drug sensitivity profiles as a function of very well-defined genetic alterations. You could profile the organoids in bulk or single cell. You could do cytokine profiles. You could do immune cell interactions, triangulated cultures that basically add stroma, as well as in vivo studies. And this is a very fertile area in the field, and we certainly are interested and well poised because we have at least the colon cancer models with the flox alleles good to go for this. And there are other commercially available types of transgenics, for example, in breast cancer, where one can do similar things. I started a company called Smurf Therapeutics. And then I'm just going to roll through this. But again, I'm not going to tell you too much about, a lot has been happening as far as the Cancer Center over the last 3 years in terms of it coming together and having programs and activities. You know, we have 3 programs. One of our program leaders, Dr. Zitkovich, is here in the audience for cancer biology. And these bring people together to talk about research directions and translational goals through our translational groups. We recruited several faculty this year, and we're very excited that they're here, and including Sheldon and Sean Lawler, and working with our colleagues, Lifespan, Stephanie Graff, Abbas, Eric Wong, and Galina Lagos. So this news just came out a couple of weeks ago that the Cancer Center is receiving a big gift and is being named the Legorreta Cancer Center. I think the official announcement from the university will be coming out next week about providing more details of this. We need space, and I'm going to have to share this with Jake on Twitter. Jake was breaking and entering, scouting out space at Lifespan that could be renovated. And we do this whenever we have some faculty we're trying to recruit, and there's a whole lot of space over there on Core 03. And, you know, he did it again. Again, looking for space. And, you know, he actually gets in through that window and opens the door to let the rest of us in. And so this is the last thing I'm going to present to you. When Eric Wong joined us here from MGH and he was at our lab meeting, you know, one of the first, I mean, he's an incredible neuro-oncologist and very interested in research and translational research, but he asked at the first meeting, do you guys have a happy hour? I said, no, we don't. You know, we're still coming out of COVID, and it's like, we can't really, but we should work on that. So then we had another lab meeting where I said, why don't we get a committee to do this? And I'm not going to volunteer anybody, but you know, it'd be nice if we have a committee to sort of organize this. So we do this once in a while. I'm not sure who's on the committee. I think it's Laura, who is the world's best assistant. Yay. This is what came out. They call it Labby Hour. And I think we have one on this coming Friday that I'm looking forward to attending. And of course, we have to give credit where credit is due. Thank you, Kim Meza, for the artwork. I believe you deserve that credit. It's very nice and looking forward to it. And I think that's all I have. So sorry for going over. We probably don't have time for questions, but I'm happy to answer questions. I'll stay because I don't have anything else for a little while, but yeah, we'll see you at happy hour. Yes. Yeah, okay. So, you know, my mentor years ago, and it wasn't just for me, you know, Vogelstein would go up to the board, like not on a board like behind the screen here, but he would go on like the wall, and he would take a marker and he would write in big letters, focus, in big black marker. The focus of our lab is on drug resistance in cancer and therapeutic combinations. And so when you talk about 30, 35 people, a bunch of those people are undergraduates and master's students and medical students. Some of them worked over the summer, they're not here. They have, so earlier when I showed you the number of papers that have come out, a bunch of these students have first author papers that have been published. But basically these days when a student comes to the lab, and it's a little bit different from what I've done at other universities in terms of getting them up to speed and working on a project. So we now have this thing called competencies, where every student in the beginning works to get up to speed with certain very basic tools and procedures and methodologies that are relevant to just about everything we do in drug discovery and development. And interestingly, some of the students who have come recently to the lab, undergraduates as well as a master's student, actually started discovering something about cell viability. We do cell viability, but how many cells you plate matters. The density of the cells matters. The observed sensitivities are different depending on exactly how you do that. And so, it's opening up a new area, but basically some of the other lab members are junior faculty who are within the space. They have their own scientists who are working with them, whether they're students or others who they have hired. And I think when you look at the group, it's really the greater group also. So we all get together, with all the new labs. So there's basically 10 faculty in that footprint up there. It's not just me doing these things. They are very synergistic, but with respect to the lab, everybody has their own project. So everybody starts out learning techniques and then differentiates based on their initial findings, based on discussions that we have. And we meet regularly. I tell the students, I want to meet with you in the beginning every week, and then later, every other week. I'm always available to speak with them. That has worked out well. We always have the lab meeting, which is very important, where people are presenting their work for more formal input. And I think, feel free to talk to the people in the lab about their experiences, what they think. It's not for everybody, because, I try to be at the bench myself. I used to be pretty good. I told you I did those 200 mini preps. I could do a lot of other things. But the one thing about me is I know how long things take. Okay, and Western blots should not take a week. If you're working with cell culture, you gotta manage the population so you can do experiments. So you don't have to wait a month because you used up all your cells, and so on and so forth. We have fun with what we do. I think we have an incredibly dedicated group of people who are very interested in what they're doing. The other thing is we have some established projects in the lab, but then when new people come, they come and they can map a path to new directions that are unique to them. By the way, that happens with undergraduates. Some have had research experiences elsewhere and know something about a certain area. They have an idea. And I think part of the fun of doing science is to have a chance to experiment, a chance to do, design an experiment and do it, see what the results are. And I'm happy to meet with people to talk, to troubleshoot or to help them, but they also talk to everybody else in the lab. It's that kind of environment. And so I think it works well. The lab has fluctuated. I learned, like, when the lab first started, within the first year of the lab's existence, we had 7 or 8 people. And it was interesting because when your lab gets to a certain size, people start to talk to each other more than they talk to the PI. And they don't always give each other good advice, okay? Like, they'll tell things and then, maybe those things work, maybe they don't work, or they'll try some new technique. And, part of the challenge for me is to find out stuff that would help people figure out why certain things are not working for them. You know, but I think it works for us. Sorry for the long-winded answer. Thank you. Oh, somebody couldn't hear me.
That might have been from a while ago.